Biological gene transgenesis in plants. The biological gene transgenesis in plants is also called vector mediated gene transgenesis, because in this gene transgenesis we used vectors for the transgenesis procedure. And also this technique is also called Agrobacterium mediated gene transgenesis. Agrobacteriums are used for the biological gene transgenesis. In this biological gene transgenesis, first we select the plasmid from Agrobacterium. Agrobacterium containing Ti plasmid. From that Agrobacterium, first we isolate Ti plasmid. The Ti plasmid means tumor-inducing plasmid. The tumor-inducing plasmid treated with restriction endonuclease enzymes. By using of restriction endonuclease enzymes, sticky ends are identified on the Ti plasmid. Later, we select one tDNA. tDNA means target DNA or desired DNA or needed DNA. The tDNA containing good characters. The good characters are developed by this gene only after transformation of gene. So the selected tDNA also treated with same restriction endonuclease enzymes. There are two pieces or shows sticky ends. Finally, the tDNA insertion into Ti plasmid with the help of DNA ligase enzyme. The DNA ligase enzyme mainly helps in ligation of Ti plasmid and tDNA. So finally, after ligation, chimeric DNA is formed. Chimeric DNA is also called rDNA. Chimeric DNA mean it contains two different types of living organism genetic material. So this portion means the tDNA is the plant genetic material and remaining portion is Agrobacterium genetic material. So two different genetic materials are present in the one form called chimeric DNA. After making of chimeric DNA, it insertion into it insertion into Agrobacterium. It insertion into Agrobacterium by using of calcium chloride solution. By using of calcium chloride solution, the Agrobacterium cell wall shows porous formation. With the help of porous formation, tDNA Ti plasmid means chimeric DNA enter into Agrobacterium through the pores. After entry of these tDNA Ti plasmid, the Agrobacterium is realized. The foreign body is entered into microorganism. Automatically, the cell wall is alerted and the porous will disappear. After insertion of tDNA Ti plasmid into Agrobacterium, these Agrobacteriums are reached to the plant cell. Initially, the Agrobacterium, tDNA Agrobacterium released their signals to plant cells. The plant cell received the Agrobacterium signals and the plant cell also released their signals to Agrobacterium. The Agrobacterium received signals through the Vir proteins. The Vir proteins mainly present in the cell wall formation. From that Vir proteins, Vir G protein important for receiving of plant signals. Finally, Vir G protein is received signals from plant cells, and the signals pass to tDNA Ti plasmid. With the signals, the tDNA involves in the transcription mechanism with the help of VIR-D1, VIR-D2 proteins. The tDNA involves in the transcription with the help of VIR-D1, VIR-D2 protein. Finally, after completion of transcription, it makes tmRNA complex. The tmRNA complex contains exons, introns. So exons are important for genetic codes. Introns are not important for genetic codes. So this type of tmRNA complex are also called immature tmRNA complex. Immature tmRNA complex reach to Agrobacterium cell wall. At this time, one channel will formed between Agrobacterium cell wall and plant cell wall with the help of VIR-B, VIR-D4, VIR-T4 single strand proteins. These all proteins are helps in formation of channel between Agrobacterium cell wall and plant cell wall. Immature tmRNA complex enter into plant cell through the channel with the help of VIR-D protein, VIR-D2 protein, VIR-E2 protein, VIR-F protein. With the help of these three proteins, immature tmRNA complex enter into plant cell cytoplasm. If the plant cell cytoplasm received tmRNA, immature tmRNA complex, it convert into mature tmRNA complex. Mature tmRNA complex means it loses their introns. Later exons are added to make mature tmRNA complex. The tmRNA, mature tmRNA complex reach to the nucleus and enter into the nucleoplasm or nucleus through the nuclear pore complex. In this function, VIR-F protein is involved. Later, mature tmRNA complex involved in the reverse transcription function to make hybrid tmRNA tDNA complex. The hybrid tmRNA tDNA complex involved in hydrolysis process. By the hydrolysis, tmRNA is hydrolyzed and tDNA, single strand tDNA is formed in the nucleoplasm. The tDNA, single strand tDNA convert into double strand tDNA with the help of DNA polymerase 3 enzyme. Later, double strand tDNA combines with histone proteins to make one T chromosome. T means target chromosome. So target chromosome containing target genes. Which gene is insertion into the Agrobacterium? The gene containing which type of genes? The tDNA genes are as it is developed in the nucleus of plant cell as a chromosome. So this process is also called indirect gene transfer technique. Indirect gene transfer technique means the selected tDNA, not insertion into plant cell. Initially, the tDNA insertion into one mediator. The mediator is called Agrobacterium. So in this biological gene transfer technique, we used one bacteria as a mediator. Agrobacterium, generally agrobacteriums are used in this method. So this process is also called Agrobacterium gene transfer techniques. Agrobacterium mediated gene transfer techniques. And also in this process, we used vectors. The vectors are important for the making of chimeric DNA. So it is also called, this process is also called vector mediated gene transfer technique in the plants.